we're going to get to the amino acids. Okay, now we're going to be talking about the third of the macromolecules, which is the proteins. So first we have to talk about what makes up proteins. Remember, we're talking about monomers first. So two monomers make up a polymer. So amino acids are the monomers of proteins. So first we need to figure out what makes up an amino acid. Okay, so an amino acid is made up of an amino group. We just learned about the functional structural formula of amino group, which is NH2. So it's just amino group NH2. Okay, hydrogen and COOH, which is carboxyl. These are all required for making up an amino acid. Okay, the R group, once again, is a side chain. It's variable. There's different types of atoms or groups that can make up this side chain. Amino acids are very, very important, and they're diverse in their range of functions, and they can serve for transport, storage, or membranes, or they may be toxins or enzymes. There's 20 different kinds of amino acids, but their functions are so diverse in range, it's incredible. Okay, but once again, amino acids are the monomers that make up proteins. Okay, but amino acid structure, what makes up an amino acid? Amino group, carboxyl group, hydrogen, and then its side chain or R group. Okay, so once again, amino acids are the monomers that make up the polymer protein. So this kind of just showing you, here's an amino acid, here's an amino acid, here's an amino acid. Once again, just like how we made a disaccharide from a monosaccharide through dehydration. Same thing here. You have an amino acid plus an amino acid. Okay, you have the dehydration. That's how you're going to actually combine these two. Same thing here. So you can have a whole bunch of amino acids, a whole big long chain of them. Okay, do dehydration synthesis, dehydration. That's actually going to bond these amino acids together to form a huge long chain of amino acids, which makes a protein. So this huge long thing right here. So right here, here's an amino acid. Here's an amino acid. Here's an amino acid. You do dehydration, that's going to actually make a protein. That's the whole point of this. These bonds, very important to learn this, especially for lab. Okay, all labs will make you understand this. This is the key, especially for using the Buret's reagent. Buret's reagent is not testing for proteins. Buret's reagent is testing for that peptide bond that's in this chain. Very, very important and key to understand, because these peptide bonds are only in proteins. Giving you guys a huge, huge key right there. Okay, so like I said, there's 20 different kinds of amino acids. Okay, these are a couple. Remember, this is what the fundamental structure of an amino acid is, that carboxyl, a hydrogen, an amino group. Okay, this is, remember, NH2, just actually looks differently the way they form it out here. Okay, but here's an amino acid, here's a different kind of amino acid. This is that R group we were talking about. Remember, it's variable, it can change. Okay, according to this R group, changes that amino acid. So there's 20 different kinds, there's 20 different forms of this that it could actually form. Okay, that huge long chain that it forms is going to be called a polypeptide. Okay, and proteins, what's the point of proteins? The point of proteins is because they're going to actually function as enzymes, or they can function as hormones. Enzymes are proteins that catalyze chemical reactions, making them happen quicker. What does that even mean? Okay, all it means is that they're going to actually make a chemical reaction quicker, happen quicker, because it just reduces that activation energy needed for that reaction to happen. So no matter what, the reaction is still going to happen. But with an enzyme, it just happens faster because the energy required, we just shorten it out just a little bit. So let's say you're pushing a boulder up a hill. Here's your boulder. Okay, this is you. You're pushing it up this hill. Okay, so your initial job is to push it up this long, big, tall hill. Okay, so you're going to require all this energy. But once you get that boulder to the top of the hill, it's all free all the way down. And no matter what, that boulder is still going to get to this bottom, and that's going to be your product, right? What an enzyme does, let's say we add an enzyme in here. Let's say we bring, great. So enzyme is going to reduce, this is the amount of energy you need to get that boulder up the hill, right? This from here to here is your activation energy without the enzyme. But we add an enzyme, so guess what? We're going to reduce the amount of activation energy you need. So now instead of getting the boulder all the way up there, you only need to get the boulder up to here because of enzyme. The enzyme reduces your activation energy required. So the same exact reaction is going to happen, but it reduces the amount of activation energy required. That's what enzymes do. That's all they do, is they reduce activation energy required. Requires less activation energy for a reaction to occur. Okay? And proteins can also function as hormones. Okay? Hormones are chemical signaling molecules secreted by an endocrine gland or a group of endocrine cells that act to control or regulate specific physiological processes such as growth, development, metabolism, and reproduction. An example is insulin. It's a protein hormone that maintains blood glucose levels. Another protein function that acts as a hormone is norepinephrine. Okay, this is actually the hormone that gives us that fight or flight reaction. It actually makes us in that sense of an emergency. It actually rushes through our system and tells us, and actually causes things such as our pupils to dilate, our heart rate to increase, our digestive system to slow down, things like that. That's what actually is causing that for us, is norepinephrine, which is a protein. And proteins, okay, have different structures. Okay, fortunately, it's very, very important for proteins to maintain their structure for it to function correctly. Okay, let's say for example this is this protein's normal structure. If it's denatured, that means it goes bleh. It's dead, it's done, it cannot reverse and go back. Okay, a denatured protein means it's been actually deformed and it is not usable anymore. That's what denatured means. Denatured protein means it's actually been conformed, or its form, it's actually the form of the protein that's been changed and it's no longer useful. Period, point blank, simple. Different things that can denature a protein are changes in its temperature. Like if you put an enzyme in temperatures that are too hot, in pHs that are too acidic or too basic, things like that completely will denature proteins or enzymes. It loses its function altogether and it's not reversible. Okay, there's 4 levels of protein structures. So if you're looking at the different amino acids, here's an amino acid. Remember, these are peptide bonds. Big long chain of them make proteins. Okay, but this sequence of amino acids, that's their primary structure. Then they can make beta pleated sheets or this alpha helix. Okay, remember this is kind of like what DNA is, just 2 of these. This is their secondary protein structures. Then you have a tertiary protein structure. Okay, It's a three-dimensional folding pattern of a protein due to side chain interactions. So now these guys, it's kind of like they kind of twist up and start reacting with itself. So it's kind of like, imagine this end comes over here and starts twisting up. That is a tertiary protein structure. And last is the quaternary protein structure, which is when the protein consisting of more than one amino acid chain. So this all the way up to here is just looking at this one. Now let's pretend like we have 3 or 4 of these. That's a quaternary structure. And our last but not least, and probably most important of all macromolecules we're going to be talking about today, is the nucleotide. Or, I'm sorry, it's the nucleic acid. But, once again, we have to talk about the monomers that make up the polymers. So, nucleotides are the monomers that make up the nucleic acids. So, nucleotides, okay, it's actually a bunch of nucleotides put together make up nucleic acids. Okay, so get that in your mind first. Nucleotide plus nucleotide, nucleic acid. Okay, so what makes up a nucleotide first? Nucleotide is made up of a nitrogenous base, a sugar, a phosphate group. Okay, it's listed right here for you. Okay, you might want to make some notes of that. Nucleotides are made up of a nitrogenous base, a pentose sugar, and a phosphate group. Nice and highlighted, and I color-coded them for you. A nitrogenous base, if its DNA is one of these 4, guanine, cytosine, adenine, or thymine. If it's RNA we're talking about, they're the same except for the last one. It's yourself, it's RNA. But the first 3 are the exact same. Okay, so key to memorize those. So if it's DNA, the nitrogenous bases are guanine, cytosine, adenine, thymine. GCAT. If it's RNA, it's GCAU. Okay? Now pentose sugar, this is also going to be different depending on if it's DNA or RNA. Okay? That's why it's called DNA or RNA. Deoxyribose, that's the sugar that's there for DNA. Deoxyribose. That's why it's called deoxyribonucleic acid. Ribose is the sugar that's there for ribonucleic acid. So that's why they got their names the way they do. Deoxyribose is the sugar here. Ribose is the sugar for RNA. Okay, and they both have the same phosphate. So like I said, a nucleotide plus another nucleotide plus another nucleotide plus another nucleotide. That's how you make a nucleic acid. Okay? A nucleic acid, what the heck is that? Okay? Well, there's 2 types of nucleic acids. What am I talking about? Okay? Nucleic acids that I'm talking about are either DNA or RNA. Okay? It's a genetic material found in all living organisms if we're talking about DNA. And RNA is mostly involved in protein synthesis. Okay, so they're very, very important that we're talking about here. So, right here is the difference in those sugars that we were just talking about with DNA and RNA. And here's a really nice listing of the differences between the 2, if we're talking about DNA or RNA. Okay, but here's the sugars. So what do you see in the difference in between the sugars? They look the same. They both have a hydrogen here. Here is where that nitrogenous base would be attached. Here's your phosphate group attached. Here's your pentose sugar. What's different? Okay, DNA is deoxygenated. Okay, deoxy. It's missing the oxygen right here. Whereas ribose has that OH right here. It has an oxygen right here. So that's how you can tell the difference. I would definitely write this down. Okay, try to memorize this. But this is what deoxyribose looks like. This is that sugar that's in DNA. Okay, it's missing this oxygen. Ribose has this oxygen right here. So deoxygenated, it's missing that oxygen. Okay, but here's the difference between these 2 as far as nucleic acids go. Okay, it's a genetic material found in all living organisms, first of all. DNA never leaves the nucleus. That's important. Highlight it, underline it, do whatever you need to do, because when we get into transcription translation, I need you guys to know it will never leave the nucleus. No matter what anybody tells you, DNA will never, ever, 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 ever leave your nucleus. Okay? DNA is a pair of anti-parallel chains of nucleotides. Like I said, it has deoxyribose as its sugar. And here are the bases that I already told you about: adenine, cytosine, guanine, and thymine. The process of DNA synthesis is replication. That's how it's made. Okay, now let's talk about RNA. Mostly involved in protein synthesis, making proteins. RNA is on the outside of the nucleus. It's a long chain of nucleotides, and it's actually involved with ribose sugar, which is right here. And the nitrogenous bases that we talked about are the exact same as DNA, except for uracil is there and is actually interchanged for thymine. And the process of RNA synthesis is called transcription. And here's an example if we want to look at DNA right here. So DNA here, right here, is how it would look if it's going down right here, because remember, it's anti-parallel, which means if it's going this way on one side, the opposite strand is going the opposite way the other way. Now, for a lab, when you guys get to transcription translation, you are going to need to set these guys up with puzzle pieces. So I want you guys to know how to do that. Okay, so right here, let's start on this side. Okay, look at this. Right here would be your phosphate. Okay, here's your sugar, and here's your nitrogenous base. So let's say this is your thymine, like for example right here. Okay, so here it is, your sugar, phosphate, and your nitrogenous base. Well, a lot of people say, well, how do I know, where am I attaching the phosphate to, or how do I know which way is going up and which way is going down, and where am I attaching each of these guys to? And how do I know which carbon is 1, 2, 3, 4, 5, and so forth? Well, let's go back one. Look at this. Wherever the oxygen is, you count to the right, and that's carbon 1. So carbon, here's oxygen, so carbon 1, 2, 3, 4, and here's carbon 5. That's how you count. Let's go back to the picture. This is how the puzzle pieces will look in your lab. So here's your phosphate, here's your sugar, and here's your nitrogenous base. So let's pretend like right here would be your oxygen. So that means, let me get a much brighter pen. Okay, here's your oxygen. So that would mean this is carbon 1, this is carbon 2. That's why this is carbon 3 labeled right here. This would be carbon 4, and this would be my carbon 5. That's where that phosphate is attached to, right here. Then the next phosphate gets attached, so carbon 3 attaches to that phosphate group, the next one, and it keeps going on from there. Here's my oxygen, my 1, my 2, here's my 3, my 4, and here's my 5. And it keeps going and going and going. Then the opposite on the other end. Okay, here's my 5 in the beginning, and it goes and goes and goes and goes. Sorry. So that's how you're going to know when you're doing that portion of the lab. So set you guys up for success right there. And you'll know where the phosphate ends on which end, and so forth and so forth. Okay? So you can see right here, the phosphate is on which end right here on the carbon 5. Phosphate's always going to be on your carbon 5. Okay? And that is all. Whew, a lot of information. I did those notes I'm telling you guys as far as for lab, those are very, very important to know. Setting you guys up for the lab to understand some things that I commonly see a lot of students have trouble with. So the keys I'm telling you guys for understanding where you want your phosphate to end and so forth, that's because I've seen those common errors or common mistakes. But any questions, go ahead and email me, and any comments or anything like that, let me know. Have a great day. Bye-bye.